Welcome to G Fun Facts Online. Today we are diving deep into a biological paradox that is reshaping how we understand the human mind. Imagine for a moment that you are a pathologist. You have two human brains sitting on the examination table in front of you. Both of these brains belong to people who lived into their 90s. You take samples from both, place them under a microscope, and what you see is terrifying. Both brains are riddled with the hallmarks of severe Alzheimer's disease. They are clogged with amyloid beta plaques and twisted tau tangles. Biologically, these two brains look identical. However, the person who owned the first brain spent their final years in a nursing home, unable to recognize their own children. The person who owned the second brain died with their memories sharp, their wit intact, and their cognitive function perfect. This is the mystery of cognitive resilience. Why does one brain succumb to the damage, while the other, facing the exact same biological assault, keeps functioning perfectly? For decades, this has been a ghost story in neurology. We knew these resilient people existed, but we didn't know how they did it. Now, thanks to groundbreaking research from late 2024 and 2025, specifically from a massive study at Boston University, we have a prime suspect. It is not a neurotransmitter. It is not a magic neuron. It is a gene called ADAMTS2. And the story of this gene is absolutely fascinating because it didn't start in the brain, it started in the skin. To understand how your brain fights off dementia, we first have to talk about collagen. ADAMTS2 acts as a pair of molecular scissors. Its official job is being a procollagen N proteinase. When your body builds collagen, which is the rope-like protein that holds your skin and bones together, it first creates a precursor called procollagen. This precursor has safety caps on the ends to prevent it from sticking together too early. ADAMTS2 is the enzyme responsible for snipping off those caps, so the collagen can weave itself into strong, durable fibrils. When this gene is mutated or broken, the result is a condition called Ehlers-Danlos syndrome, type 7C. In animals, it is called dermatosparaxis. The skin of these individuals is incredibly soft, velvety, and tragically fragile. It tears like wet tissue paper because the structural ropes were never tied correctly. So, for years, geneticists boxed ADAMTS2 in as a skin gene, a structural gene. But biology rarely wastes a good tool. It turns out that the same molecular scissors that strengthen your skin are secretly the architects of your mind. The major shift in our understanding came when neuroscientists stopped looking at neurons as the only things that matter. For a century, we viewed the brain like a computer, focusing only on the wires and the electricity. We ignored the glue holding it all together. This glue is the extracellular matrix, or ECM. It is not just inert scaffolding, it is a dynamic, bioactive environment. It is like a memory foam that instructs neurons where to sit, when to grow, and how to survive. This is where ADAMTS2 enters the neurological stage. In that landmark study at Boston University, researchers did something crucial. They looked for the genetic secret of resilience, specifically in African American brain tissue, a population that has been historically overlooked in Alzheimer's research. When they compared the brains of people with dementia against the resilience, those who had the plaques but not the symptoms, ADAMTS2 was the #1 gene that was different. It was significantly upregulated in the resilient group. This finding was then replicated in European populations, proving it is a universal human mechanism. This implies that when a resilient brain senses the toxic attack of Alzheimer's, it cranks up the production of ADAMTS2. It uses this gene as a shield. But how? How does a collagen-snipping enzyme stop dementia? The research points to three distinct, sophisticated mechanisms. The first is vascular integrity and brain stiffness. The brain is soft, but it is fed by a rigid network of blood vessels. As we age, and specifically in Alzheimer's, these vessels get coated in amyloid plaque, a condition called cerebral amyloid angiopathy. This makes them brittle and prone to leaking. The theory is that ADAMTS2, by strictly regulating the collagen in the basement membranes of these blood vessels, reinforces the pipes. It maintains the perfect mechanical tone of the neurovascular unit. If you have high levels of this enzyme, your blood vessels might be strong enough to withstand the amyloid buildup without bursting or leaking toxins into the brain. This allows your brain to keep clearing out waste through the glymphatic system, which runs along these vessels. Essentially, ADAMTS2 keeps the plumbing working even when the pipes are old. The second mechanism is even more elegant, and it involves a protein called reelin. Reelin is a superstar in neurology. During development, it tells neurons where to go. In the adult brain, it strengthens synapses to encode long-term memory. You might think that more reelin is always better. But in a diseased brain, signaling pathways can get noisy. Chronic inflammation causes signals to fire randomly. Recent biochemical studies show that ADAMTS2 acts as a specific inactivator of renin. It cuts the protein to shut the signal off. This seems counterintuitive. Why would a resilience gene destroy a memory protein? The answer is likely about signal-to-noise ratio. By precisely cleaving renin, ADAMTS2 prevents the system from becoming saturated. It ensures that the memory signal is crisp and temporal, happening only when needed, rather than flooding the system with noise. It brings order to the chaos of a degenerating brain. The third mechanism involves the immune system. ADAMTS2 has been shown to remodel the matrix to release a stored chemical called TGF-beta. TGF-beta is a master cytokine that is neuroprotective and anti-inflammatory. By digging this chemical out of the matrix, ADAMTS2 acts as an immunological thermostat, suppressing the brain inflammation that usually drives cognitive loss. So in the aging brain, ADAMTS2 is a vascular reinforcer, a signal tuner, and an inflammation suppressor. But the story gets even stranger, because this gene is also controlling the very architecture of the brain before you are even born. This takes us to the realm of brain development. The human cortex is a 6-layered marvel. To build it, neurons have to travel from deep inside the brain to the outer surface, a process called radial migration. When neurons are born, they are shaped like confused stars. They are multipolar. To move, they have to transform into a streamlined arrow shape. They must become bipolar. Research from 2022 and 2024 revealed that ADAMTS2 is the trigger for this transformation. As neurons reach the waiting room of the brain, known as the subplate, they secrete ADAMTS2. This remodels the matrix around them, giving them the signal to change shape and start running. Without this gene, neurons get stuck. They pile up in the deep layers, leading to cortical dysplasia. This is critical because these kinds of migration errors are often the root cause of epilepsy, intellectual disability, and schizophrenia. Speaking of schizophrenia, the link here is profound. ADAMTS2 is not just a builder, it is responsive to the brain's chemistry. Its expression is regulated by dopamine D1 receptors. In schizophrenia, where dopamine signaling is chaotic, ADAMTS2 levels are often abnormal. Interestingly, when patients are successfully treated with antipsychotics, their ADAMTS2 levels return to normal. This suggests that the gene is part of the machinery that maintains the cognitive reserve in psychiatric patients, buffering their minds against the disorder. We also see this architectural role in pediatric stroke. Stroke is usually an old person's disease, but when it happens in children, it is often a genetic failure of the vessels themselves. Genome-wide association studies have flagged ADAMTS2 as a top susceptibility gene for pediatric stroke. This brings us full circle back to the skin. Just as mutations cause fragile skin in Ehlers-Danlos syndrome, subtle variations in this gene can cause fragile blood vessels in the brain, making them prone to tearing or clotting under stress. So what does all of this mean for the future of medicine? The discovery of ADAMTS2 as a resilience factor opens a new frontier called matrix therapy. For 30 years, we have tried to cure Alzheimer's by attacking the plaque, with very limited success. This new paradigm suggests a different approach. Instead of just trying to clean up the trash, we should focus on reinforcing the building. We could potentially develop gene therapies that deliver ADAMTS2 specifically to the hippocampus of patients with early Alzheimer's. Or we could create small molecule drugs that stabilize the enzyme. The goal would be to induce that state of synthetic resilience. We might not be able to stop the aging process or the accumulation of plaque, but if we can boost the expression of ADAMTS2, we might be able to arm the brain to function perfectly despite the pathology. We could turn vulnerable patients into resilience. Furthermore, this gives us a powerful new biomarker. In the future, a doctor might test your spinal fluid not just for Alzheimer's risk, but for your resilience factor. If you have high amyloid, but high ADAMTS2, the prognosis changes completely. It moves from a sentence of doom to a manageable condition. The story of ADAMTS2 teaches us a fundamental lesson about biology. It teaches us that neurology is not just about electricity, it is about architecture. The soft machinery of the extracellular matrix is the bedrock upon which our memories, our sanity, and our longevity rest. We have spent decades obsessed with the neurons, but the secret to saving our minds might lie in the space between them. That is the power of the hidden architect, ADAMTS2.